Good evening, everyone, and welcome to the second February Craw lecture. I'm George Craw. This series features acclaimed UC Santa Cruz scientists and technologists who are grappling with some of the biggest questions of our time. Tonight's speakers, Professor Raphe Kudela and Associate Professor Kristy Kroeker. Mark received a bachelor's degree in biology from UC Santa Cruz in 1976, a master's degree from San Francisco State University in 1983, and a PhD from UC Santa Barbara in 1991. The purpose behind Mark's research is to better understand the structure and dynamics of populations and communities of nearshore marine organisms and their ecosystems. Before we begin, I would like to share a few details about the event tonight. We are using a webinar tool, so there is no chat function. We will have an opportunity to answer your questions at the end of the program, and we invite you to submit your questions into the Q&A box at any time. Tonight's event will be recorded and be available online in a couple of days. Now I'll turn this over to Mark.
Thank you, George, and thank you and your Raphe for sponsoring this great lecture series. It's my great pleasure to introduce our two speakers this evening, but before I do, I wanted to quickly reiterate that we're going to hear from both speakers before moving to the question and answer period. So please hold your questions and hopefully we'll get to all of those after the two talks. Dr. Raphe Kudela is a phytoplankton ecologist and professor and chair of the Ocean Sciences Department at UC Santa Cruz. He also runs UCSC's Biological and Satellite Oceanography Lab. Raphe received his bachelor's degree in biology and marine sciences at Drake University and a PhD in biological sciences at the University of Southern California. His research interests include ecological modeling and remote sensing, satellite oceanography, phytoplankton ecology, harmful algal blooms, and answering the fundamental question, what controls phytoplankton production and distribution in the ocean? Dr. Kristy Kroeker is an associate professor of ecology and evolutionary biology and runs the Kroeker Ocean Sustainability Lab here at UCSC. Kristy is a Packard Fellow in Science and Engineering and a Sloan Fellow in Ocean Sciences. She's motivated by a fundamental interest in the drivers of change in marine communities and a desire to improve our ability to forecast and manage the effects of environmental change in dynamic, complex marine ecosystems. This interest is reflected in the basic research of her lab, but she's also very interested in coastal sustainability policy and management. So welcome Raphe and Kristy, and without further ado, I'll turn it over to Raphe.
All right, thanks so much, Mark. And I'd like to extend my thanks as well to George and Raphe Craw for hosting this wonderful series and giving us an opportunity to speak to all of you today. And so I'm going to bring up my slides and there it goes. All right. And so I'm going to start by telling you a little bit about what climate change is doing in our backyard, the California current system. And so before I get too far in, I want to acknowledge a couple of key people that are essentially co-authors on this talk. Meredith McPherson is my PhD student, and she's done a lot of the work that I'm going to present on kelp. And Dennis Finger was an undergraduate student at UC Berkeley who was in a NASA summer program with me and also did a lot of the groundbreaking work on the kelp forest ecology material that I'll show. And so starting with this first image in the upper right, this is a temperature anomaly. And so reds and yellows are where it's warmer than normal in January of this year. Blues are where it's cooler than normal. And I highlighted some things that I'm sure everybody's heard about. We have a La Nina right now going on. And so cooler than normal conditions in the equatorial Pacific, which is of course the opposite of an El Nino. We've got the polar vortex, which is freezing Texas or was freezing Texas. And then we've got ongoing heat waves in the upper part of the Pacific. And I bring this up because it's a really good example of the fact that the ocean has its own rhythm. We have seasonality in the ocean. We also have changes on interannual scales, so years, but also decades. And so El Nino is a really good example of something that occurs on decadal timescales. And they're all actually connected. And so the La Nina, for example, helped to steer the Gulf Stream, which resulted in the polar vortex extending so far into the continental United States. But what I really want to focus on today is how some of these factors, these rhythms in the ocean, affect the biological organisms of the ocean. And in particular, I'm going to start by talking about harmful algal blooms. And so that figure to the bottom left, this sort of cartoony looking one, that's depicting the area that was impacted by the massive domoic acid poisoning event in 2015 that shut down the Dungeness crab fishery. And that was the largest bloom of that kind we've ever seen in the world, certainly the largest in California. And one of the questions we get asked a lot was, is that climate change? The image on the right, of course, is one of our iconic kelp forests. And as you may know, we've seen massive die-offs of kelp in Northern California. And again, the question comes up, well, is that climate change? And we have to keep in mind that there are natural patterns. And so El Niños and La Niñas come and go. The polar vortex is not something that showed up with climate change. And the question is, can we actually separate natural variability from these anthropogenic forcings that humans are causing? And so one of those really obvious forcings, and this is a beautiful but disturbing image, shows the warm anomaly, the so-called warm blob that occurred starting in 2014 and extended into 2015 and ended up triggering the Dungeness crab shutdown. And you can see here really, really warm conditions. And so that sort of red bullseye is much, much, much warmer than normal. And I'm going to introduce some terms that we use when we talk about climate change. This figure is from a recent IPCC report on the oceans and cryosphere. And I'm going to highlight just a couple of them. I know there's a lot of panels there. I'm going to pull out a few things specifically. And so if we start with that panel in the upper right, what we're looking at is change over time. And then in the left, I'm showing you how we actually define a marine heat wave. And so a marine heat wave may be a new term that you haven't heard a lot before. It's something that has really emerged with climate change in the last 10 years or so. And a marine heat wave is a lot like a land heat wave. It's above normal temperatures for a prolonged period, for longer than 5 days in the ocean. And so generally that above normal, we reference it compared to what we would expect to see in a given year. And it has to be in the top 10% of what we expect. And so statistically, a very unusual event. And so again, 2014, 2015, massive marine heat wave, definitely above normal. If we go to that figure in the upper right, the way we calculate that is we look at some baseline period. And so for climate change, we oftentimes start with the Industrial Revolution, and we go from there and we say, well, has it changed compared to before humans really started pumping carbon dioxide into the atmosphere? And so what we're looking for is when that natural variability, the noise, exceeds the threshold. And we can say, at that point, it really is climate change because it's beyond any variability that we've seen in the past. And so if we go back to our marine heat wave, this is what it looks like going from 1850, from when the Industrial Revolution really kicked in and we started adding carbon dioxide to the atmosphere, up to present day. And so our present day is roughly sort of right here, and then forward to the end of the century, to 2100. And what you're looking at is the recorded number of marine heat wave days up till today, and then the projected number of marine heat wave days in the future. And what I want you to just notice is how similar these two graphs are. This is a beautiful example of an increase in the ocean that's being directly attributed to climate change. The ocean is warming, we're getting more marine heat waves. We've gone from basically almost no marine heat waves for the last 150 years to a significant number, and it's projected to just keep increasing until we reach a threshold in 2100. The different colors on there, reds and yellows, are the different model projections that we use. And so this is an example where what we're doing is we're saying, do I feel confident that the number of heat waves we're having is more than I would have expected without climate change? And so that's a key thing that we want to keep in mind, is that when we talk about, is this climate change or is it not climate change, often we can't say with 100% certainty that it is or it isn't, but we can say statistically it's more likely than not to be climate change. And that leads us to the bottom right. And the way we often do that is we use statistics and we say, well, let's look at the real world, what's going on, and then compare that to these model runs. And if we can't predict the variability in the real world without adding human activity, then we can do something called attribution. We can say that's attributed to humans and climate change. And that's really, the key thing for climate scientists is not just to document that there's more marine heat waves or that there's kelp forest die-offs, but actually to be able to say with confidence that this is being caused by climate change. And so one of the things that there's been a lot of discussion about is one of the things that we expect might happen with climate change is there's going to be biological responses. One of those responses is more harmful algal blooms. And we see this all over the world. This figure is from a recent paper that came out about a year ago, a little over a year ago. And it's highlighting that we see big blooms of Pseudonychia that produces domoic acid in our backyard in California. But in the same time period, we've seen more paralytic shellfish poisoning caused by this organism called Alexandrium in Tasmania, where they've also been experiencing extreme marine heat waves. And we've been seeing the same organism, Alexandrium, and another one called Pseudochatnella that caused massive die-offs of the farmed salmon down in Chile a few years ago. Each one of these events has been linked to climate change, that these organisms do better in a warm climate and that they're responding to the things that we're doing. And so these are really good examples of some of these other patterns that are in the IPCC report. And when we get into biology, it's often not linear. And so as temperature goes up, we don't just simply get more of a bloom or less of some other organism. Instead, we can reach these things we call tipping points, where you fundamentally shift the ecosystem from one state to another state. And that's related to the bottom one, cascading effects, where it's often difficult to predict what's going to happen with the biology. And so we know from just basic lab principles and physiology that as you raise the temperature, we speed up metabolism, we do other things, but there's these unknowns that pop out of the real world that are not necessarily predictable. And so we've got multiple levers being pulled at the same time, and climate change is causing these forcings, and we don't necessarily know what's going to happen. And so that's both exciting and interesting if you're a scientist, and also, of course, a little bit scary because it means that we can tell you it's going to get warmer, but we don't necessarily know what's going to happen with the biology. And so I'm going to jump in and tell you how we're trying to tease apart what's going to happen with the biology based on looking at the past and looking at the data that we've generated so far. And so we're going to focus on, again, this organism Pseudonychia, which is a diatom. Kind of unusual. Mostly diatoms are really good for the environment. They feed fish, they drive fisheries. They're generally considered part of a healthy ecosystem. But this one produces domoic acid. Domoic acid is a toxin that basically overstimulates your nervous system. And so if you live here in California and you've ever seen a sea lion that's very disoriented and seizuring, that's usually domoic acid poisoning. We've had really unusual events where birds have literally fallen out of the sky because they've been poisoned by these toxic algae, and it's enough that they just can't even fly anymore and get completely disoriented. Well, one of the things that we like to do is instead of just using models, we really like data, that you can look at the real world and say, what does the world tell us about what's changing and how these changes are going to influence things? And so this graph on the bottom is a summary of more than 10,000 individual measurements of where we have domoic acid in the California current. And with that, we can measure temperature and salinity and nutrients and other things. And then we can say, well, are there patterns that come out from this massive data set? And so this data set went from the year 2000 to the year 2020, so 2 decades of data. It includes El Niños, marine heat waves, cold years, warm years, low productivity years, high productivity years. And so it's a big data set. And what we're looking at is the y-axis. It goes from 0 to 1, and it says how likely would it be that if the cells were present, you would see the toxin at the same time. And we've plotted it as a function of temperature. And so we used a particular statistic that can pull that out. And what you can see is we're more likely to have toxin at a particular temperature range. And so this peak right there is about 13 degrees Celsius. And so that's our typical temperature in Monterey Bay, for example, which is why we tend to get a lot of toxic blooms here. If we cool the ocean off, you see it drops down a little bit. And so they don't do so well in really cold conditions. And if we warm the ocean, it drops down really fast until we get to about 20 degrees. And then with 10,000 observations, we just don't see toxin when it's 20 degrees or warmer in the ocean. For those of you that are interested in the details, that sort of the black solid line is the average, and then that gray envelope around it is the standard deviation. And so this is a really tight relationship. And what that tells us is that temperature turns out to be a really critical driver. You do need the cells there, so there's a lot of biology going on behind that to explain why we get pseudonychia in the ocean and at certain times of the year. But if the temperature isn't right, you're not going to get toxin. And so we can take that information and put that into some of our other models. And we can basically say, well, it's a little bit like Goldilocks. If it's too cold, Pseudonychia doesn't like it. They're not going to produce a lot of domoic acid. If it's too hot, Pseudonychia doesn't like it. And even though the cells can keep growing, they are actually no longer producing toxin. And if we're in that sweet spot where it's not too cold and not too hot, we're really likely to produce a lot of toxin. And so what we've done is we've now gone into the ocean and we've said, okay, well, temperature is clearly this key driver. And so here what I'm showing you is the west coast of the United States going up into the Gulf of Alaska and down a little bit south of Baja, Mexico. And we're looking at reality. And so this is a satellite temperature product called GRIST. You don't need to worry too much about what that is, but it's an average temperature for June 2015. And I've used that sort of stoplight color coding to show you where we would expect Pseudonychia to do well. And so green, it's in that sort of optimal temperature. Yellow, it's getting a little warm. Red, we would expect no toxicity at all because it's actually too warm for it. And then the small blue areas you see would be where we think it's probably too cold to get a lot of toxicity. And so this is what it looked like in June 2015 when we had that massive bloom. And you can see that green area lines up really, really well with the area where we saw all this toxin showing up. And so it was really dominant from about the Channel Islands, and it extended all the way up into the Gulf of Alaska. And that was extremely unusual. We normally see this, these blooms in California. They had never seen toxicity in the Gulf of Alaska before. And so something new that had popped out of many decades of observations. And so this is reality. This is what the temperature actually looked like. And then I just simply color coded it by this temperature function. And then what we can do is we can take these models that are climate models that project into the future. And what's really nice about this is the models that most of the climate scientists have been using is these models called CMIP5. And so there's these climate models that model the entire world at the same time. And they project out to 2100 what it's going to look like. But these models started running back in 2010. And so now, of course, it's 2021, which means we can look at the first 10 or 11 years of that model and see, was it accurate or not? Did it do a good job of showing what we know actually happened? And so this is the model data for June 2015. And a couple of things to point out, you'll notice it's not quite as detailed, and that's because the model resolution is at about a degree resolution and the satellite resolution is at about 4 kilometers. And so there's a little bit of mismatch in scale, but you can see the patterns of the color look pretty similar. And that's actually pretty impressive given that when this model was run, it had been running without any input for 5 years. And so this was a projection that had been running for 5 years, and it still captures what we actually measured in June of 2015. And so it gives us confidence that, well, if it was correct in 2015, we can now go forward in time and say, where might we expect to see Pseudonychia blooms and where might we expect to see a lot lot of toxicity. And so we did exactly that. And so here's 2030 on the left, 2050, the middle of the century in the middle, and then 2080 getting close to the end of the century on the right. And there's some really obvious things that are going on. The green is getting pushed further and further north. And so the Pseudonychia is essentially following the temperature that they like, and it's moving up into the Gulf of Alaska. And so for us that live here in California, that may be a good thing, that we are actually sort of in this yellow region within the next 10 years or so. And so we would expect that it's actually getting too warm to produce lots of toxin. But of course, it's a really bad thing for the Gulf of Alaska fisheries, because they're suddenly going to have a problem that they've never really had before, that's going to be the perfect conditions for these bloom organisms to be producing the toxin. And of course, you've got to be wondering, well, if Pseudonychia is moving north, what about something that's further south of us? And so one of our big concerns is that as Pseudonychia gets pushed further and further north, something that likes much more tropical waters is going to come up into our area. And instead of dealing with a problem that at least we understand, we're going to be dealing with a problem that we've never seen before. And of course, a big question with any of these projections is this really going to happen? Do we actually believe these models? And so we've started to see some evidence that this is in fact happening. And so I'm going to show you an example from this paper that we just very recently published. This is in Frontiers in Climate. And what we've seen is that we're seeing exactly what we predicted, that we're actually seeing the blooms and the high toxicity moving northward and showing up in new locations where it never was before. And so if I just walk you through this, on the left, panel A and panel B are the average sea surface temperature in springtime, when we mostly get these blooms, between 2002 and 2013, before we had all these marine heat waves, and then 2014 to 2019, after we've had all these marine heat waves. And so you may have heard about the big warm blob that we had in 2015, but we've actually had a continuous series of marine heat waves since then. And so it stayed warm in the California current. And so panel C is just simply subtracting one from the other. And what you can see is everywhere you see warm colors, which is basically the whole figure, it's much warmer than it was before 2013. And we're talking pretty significant. And so the reds and the orange colors, on average, we're getting more than 1 degree Celsius of warming. That's a big, big impact on the coastal ocean. D and E, now we're looking at what does that do to the biology? And so same time period, D is 2002 to 2013, and then E is 2014 to 2019. But now we're looking at the presence of toxin. The blues and purples mean that basically we didn't see toxin there. And this great big bullseye that you see there, that sort of red, that's basically off Humboldt Bay, Trinidad headline, if you're familiar with that. And this has emerged as a new domoic acid hotspot that showed up after the marine heat waves, and it's been persistent. It's still there today. And we've been able to link that to actually attribute it to climate change, that the temperature is definitely attributable to climate change. And the fact that we simply did not see toxin in this area until after it warmed up, we think we can also attribute this biological response to it. And so we've basically generated a new hotspot for domoic acid. If you know anything about the Dungeness crab industry, that's also very worrisome, because that is a big Dungeness crab area. And so we would expect going into the future that we're going to have more and more of these problems. And so this is a really good example of a tipping point where we've fundamentally changed the ecosystem. We went from one ecosystem where we didn't really have a lot of toxin in that area to our new reality where we're getting a lot of toxin in that area. And so that's the harmful algal bloom side. And when we get to questions, I'd be happy to answer any additional questions about that. But I also want to turn and tell you what's happening to kelp, because kelp is another really interesting story that's responding to these marine heat waves and responding to climate change. But instead of actually warming up and making it better for them, it's warming up and making it worse for them. And so what we've seen is that there's this massive loss of kelp in the northern kelp forests. And so the way we track that is the traditional way is you go out and do diver surveys. And so Kristy and Mark and researchers like that, they spend their careers doing that. They go out and physically measure the kelp. They physically look at the organisms that are in there. But it gives you sort of a patchy view of things. California Department of Fish and Wildlife also measures it by flying over in an airplane and taking imagery, and then they digitize that and they can calculate the area, but it's expensive. And so for many years, they basically just can't afford to fly the planes. And so Dennis Finger, this undergraduate student, he came up with this method where we can use this satellite called Landsat, and we can go back all the way to 1985 and then forward until the satellite stops working, and it's still working today. And we can generate this long, consistent time series. And so what you're looking at is this box and this dashed line between Point Arena and Jenner, this area that's dominated by a particular type of kelp called bull kelp, which you may be familiar with. And on the left, we have 2008, 2009, 2014, 15, and 16. The green is where the satellite tells us there should be kelp. The sort of gray is where the California Department of Fish and Wildlife surveys said there was kelp using their airborne flights. And then where you don't see any green or gray means the kelp is gone. And so you can just see this dramatic decrease from 2008 before the marine heat waves to 2016 after the marine heat waves. And all that kelp is just getting decimated. And so one of the things we were able to show is that statistically we do as well as CDFW. And so now we can generate this multi-decadal record of when there's kelp and compare that to other factors like temperature, like wind waves, and like nutrient runoff and things like that. And when we do that, instead of looking at just that square, this is from Meredith McPherson's work. Now we're looking at the whole West Coast United States, and everywhere you see green means there's kelp, and where it's darker green, it means there's more kelp in a particular year. And there's a couple of things to notice. There's sort of a band of gray right here. And that's because kelp doesn't grow in that part of the coastline because it doesn't have the right benthic substrate. And so it can't attach to anything. And so we don't expect kelp there. But what I'd like you to notice is kelp comes and goes. And so there's this, again, this natural rhythm to the ocean. The kelp forests are sometimes healthier, sometimes less healthy. Sometimes it extends further north, sometimes not. But when we get to the end of the record from 2015 on, basically the right side of this figure, you can see that the green has gone away. That's the loss of these kelp forests in Northern California. And so now we have the data to say, well, we know when there was kelp and when there wasn't kelp. What does that have to do with temperature? And so here we're looking at, again, on the left, 2008 versus 2019. In this one, the kelp is shaded in this sort of orange color. And so this is where we would normally have kelp. And you can see in 2019, there is no kelp, it's gone. And so we can take this long data set and start querying it and saying, well, what do marine heat waves and temperature have to do with that? And so on the right, you'll see these figures that looked a little bit like that one I showed you earlier, and I'll pull that up again. And basically what we're doing is we're making statistical histograms. And so is this normal or not normal? And we can divide the years up into years where we had El Nino, which is shaded yellow, normal years where there's no shading at all, it's just white, and then heat waves where it's either pink or dark orange. And so dark orange is where you have an El Nino and a heat wave together. Pink is where you just have a heat wave. And then down here in the bottom where you see the green bars, this is the kelp canopy area with those El Ninos and heat waves overlaid on it. And what I'd like to point out is we have had big El Ninos in the past. And so there was a big El Nino in 1997, 98, but it didn't make all the kelp go away. When we get to the end of that, so 2014, 15, 16 into 2020, we had a combination of an El Nino and a marine heat wave, and it really, really decimated the kelp. And so clearly temperature is doing something, but it turns out that it's not temperature. And remember, I told you that when we get into biology, it's messy, it's not linear. We get into these tipping points and other things going on. And so Meredith McPherson's work kind of dissected that a little bit. I'm going to give you sort of a high level example of that. she developed a model to try to predict how much kelp you would expect based on a bunch of environmental parameters. And so starting in the bottom, those green bar plots again, that's kelp area, but now we've turned it into a statistical change. And so above zero, more kelp than on average, below zero, less kelp than on average. And so again, we're seeing this sort of this rhythm that sometimes kelp is doing better, sometimes it's doing worse. These two boxes on the left, she tried to model it using just physical variables. And so you don't need to worry too much about it, but we looked at nitrate concentrations, the number of marine heat wave days, when we had spring transition to spring temperatures, wave height, and then these decadal oscillations like the Pacific Decadal Oscillation and the El Nino. And she does a reasonable job of predicting it. And so the black line down here that's overlaying the green is the model output, but we're only predicting about 28% of the variability. And so we can get the sort of large scale pattern, but we're not doing super great at predicting when kelp would be coming and going. A key thing that we failed on is if you get to the end of the record, if we only use these physical variables, we would predict that the kelp would recover, that the kelp would have come back in 2017, 18, 19, 20. Instead, there's no kelp. And so clearly just these physical drivers like temperature are not explaining what's going on. And so Meredith added in data from a bunch of collaborators, including Mark Carr, who's on this paper, and brought in purple urchins and also sea stars. And our predictability went up to about 52%. And for predicting biology, that's actually quite good. And the key thing, if you look towards the end of this record in panel D, you'll see that here's the model and here's the prediction. And it predicts that when you add in urchins, that basically we've turned Northern California into an urchin barren, that the urchins are grazing down the kelp and the kelp haven't had an opportunity to recover, even though the marine heat wave has kind of relaxed. And so what we see is temperature is absolutely critical, that the temperature is driving a lot of this, but it's the temperature combined with the biology that's keeping our iconic kelp forest from recovering. And so again, similar to the HABs, we've reached a tipping point. The combination of the predators and the temperature and the environmental conditions have shifted us from nice, healthy kelp ecosystems that have recovered from El Ninos in the past to urchin barrens, where we don't have any kelp and we have way too many purple urchins, and we've lost all of that biodiversity and all of the wonderful things that kelp do for us, including pulling down carbon dioxide and helping to mitigate climate change. And so just to wrap up my part of it, there's going to be winners and losers with climate change. One of the winners, we think, are going to be harmful algal blooms, unfortunately for us, that as that temperature is shifting, because of climate change, that we know absolutely that this temperature is well beyond anything that would be caused by El Nino or natural changes or Pacific Decadal Oscillation, that there's a clear link between that change in temperature and the harmful algal blooms, that they produce more toxin in a certain temperature range, and they're simply moving around with that optimal temperature window. And so more and more we're seeing high toxicity further north of us, but we still see it here in California as well, because we're still in that optimal temperature range right now. The losers, unfortunately, in this scenario are the kelp forest ecosystems. They're really getting hammered by this combination of high temperature and the tipping point that's being caused by an overpopulation of urchins, because the sea stars, due to sea star wasting disease, have been decimated as well. And so the natural predator of the urchin has gone away. And so what we see is that temperature really is critical, but the loss of resilience of these ecosystems is more complicated than just simply looking at temperature. And so since GameStop and the stock market has been in the news a lot lately, I thought I'd end with something that you hear a lot if you're in the stock market. Past performance is not indicative of future results. And so the caveat to all of this is we can look backward and we're pretty confident that we know what happened, and we're pretty happy with our models. We can run those models forward to today and we could look to see, did they actually predict what we would expect over the last few years? And they are. but we don't know for sure that we can actually predict what's going to happen in 2100. We can tell you absolutely temperature is going to go up and there are going to be consequences. But some of the things that we don't know is, is something going to change and the kelp forest recover? Is something going to change and the toxic algae end up going away? And something that we're working on right now, and you'll have to stay tuned because we don't have the answer yet, but we're running an experiment with the toxic algae in our lab right now, where we're growing them at that higher temperature for 1,000 generations to see if they actually evolve. And if they evolve, are they going to be more sensitive or less sensitive to temperature? And so those types of experiments combined with these climate predictions can give us a handle on who's going to be the winners and who's going to be the losers going forward into the future. And so with that, I'll stop there and I'll turn it over to Kristy. But I would like to acknowledge a lot of this work has been funded by NOAA and NASA, and here in our area, the Central and Northern California Ocean Observing System. And so I'd just like to acknowledge where all of that data and inspiration came from. So thanks very much. And when we get to questions and answers, I'd be happy to follow up on any of that.
My apologies, just working on my screen sharing. So good evening. Thank you all for being here and sticking around. So thanks, Raphe, for an excellent talk. It's really interesting to actually see what's been happening on the coast in the last couple of years. And I also wanted to thank George and Raphe for hosting the seminar series and giving me an opportunity to speak to all of you about my research, as well as all of you out there for hanging out on a Wednesday night for an online seminar series. So my research at UC Santa Cruz is focused at this intersection of community ecology, environmental change, and policy and management. And my intention of working at this intersection is really to inform sustainable management of our ecosystems in the future. So I'm in the Ecology and Evolutionary Biology department, and as a community ecologist, this is kind of my view of the world. I've always been interested in what happens at really local scales to affect these individual organisms actually scale up to the patterns that we see in nature. So what happens to this fish in this dynamic functioning ecosystem? Now, when I think about environmental change in the ocean, it's really threefold. So I'm thinking about the kinds of changes associated with anthropogenic carbon dioxide emissions. The oceans have absorbed over 90% of the heat associated with global warming. And Raphe just did a great job talking about some of the repercussions of that warming. At the same time, another repercussion is that warmer ocean water can hold less oxygen. So we're seeing this global process of deoxygenation happening worldwide. And then furthermore, the oceans have actually absorbed about 1/4 of the carbon dioxide that we've emitted since the Industrial Revolution. This leads to really predictable changes in the carbonate chemistry of the ocean in a process called ocean acidification. So the research in my lab is really focused on trying to understand the emergent effects, not just for the fish, but for the entire ecosystem to this range of changes that we're seeing in the ocean. However, for today's talk, I'm really just going to focus on the acidification piece, primarily to complement what Raphe has been talking about. And I'm going to be focusing on kelp forest ecosystems as well. So I'm going to talk about 3 pieces here. I'm going to start by giving you just a little bit more background on ocean acidification itself. What is it? Why should you care about it? Then I'm going to transition to a little bit farther up the coast in Southeast Alaska. And I'm going to tell you about some research we've been doing with this really culturally important species, the Pinto Abalone, where we've seen that really seasonal fluctuations in the chemistry mark these windows of vulnerability to ocean acidification. And then last, I'm going to talk about some research that's being led by one of my graduate students, Emily Donham, here on the California coast, where she's been looking at how ocean acidification is going to show up in California and how that might affect this really important grazer. Okay, so starting with ocean acidification, what is it, why you might care? So as I mentioned, the oceans have absorbed about 1/4 of the carbon dioxide that we've emitted. This is good for global warming. It has actually slowed that process, but it causes these really predictable changes in chemistry. So I'm showing you 2 figures here, one from the late 1800s and then one from 1995. These are model results where warmer colors represent lower pH or more acidic waters. So you can see since the Industrial Revolution, the pH has decreased about 0.1 units. So the whole ocean's gotten a little bit lighter. Now, as we look ahead to the end of the century, so now I'm showing you that 1995 figure on the left, but 2095 on the right, we expect about 100% increase in the acidity of the global ocean. Now, this is a big deal for an ocean that typically has very, very stable temperature and chemistry. The things I want you to get from this figure is, in part, the scale of the issue that we're looking at, but also that it's happening everywhere, that this is going to affect every species and ecosystem in the ocean. While it's a global threat, there are also hotspots. So one thing you might see is those warmer colors up at the northern portions of the figure. So we expect these polar waters, both in the northern and the southern hemisphere, to actually acidify more rapidly because cold water can hold more carbon dioxide. In addition, we are in a hotspot here in the California current. So these 2 figures are the West Coast, and I've shown you Monterey Bay there. I've tried to kind of point that out. For the year 2005 and the year 2050, again, these are modeled figures. The color bar is actually quite similar to the previous figures. Warmer colors represent more acidic conditions. And what I want you to see here is by mid-century, so 2050, so 30 years from now, we're expecting to see pH values or acidity conditions that we're not expecting to see in other areas of the global ocean until the end of the century. So we're really accelerated here in California. Now, the reason for this is really due to our local oceanography. So I'm going to just take a minute so you can understand this. And it's driven by this process of upwelling. So those of you who are in California right now have experienced the upwelling winds in the recent days. What happens is that the earth warms up a little bit faster than the ocean, and you get these longshore breezes that blow down the coast. As those breezes blow down the coast, they actually push the surface waters of the ocean down the coast as well. As the earth spins, the Coriolis effect actually forces those waters offshore. And those surface waters are then replaced by these deeper waters. Those deeper waters tend to have higher nutrients, as well as higher carbon dioxide and lower oxygen conditions. So that's a normal process. Upwelling is really important. It's what fuels our ecosystems. It's why we have kelp forests here in California. However, those waters, those upwelled waters, are becoming more acidic because of this global process of acidification. What's happening is the carbon dioxide is actually, it dissolves into the ocean at the surface, but it's used by phytoplankton, which are eaten by other species, or they die. Either way, as carbon makes its way through the chain, those carbon molecules sink to the bottom of the ocean. And those organisms that ate up that carbon in the first place get broken down, and that carbon dioxide is released. In that same process, oxygen is used up. And so we're taking that carbon dioxide that's entering the ocean through acidification, and we're concentrating it down in the depths of the ocean. So we're really accelerating the process here in California. Okay, now this has already showed up. So this is a headline from the Seattle Times, actually a number of years ago now. And what's really interesting here is through some really productive partnerships between scientists and industry in the oyster or shellfish aquaculture industry, we were able to show that these massive die-offs at oyster hatcheries in Oregon and Washington could actually be linked to these more acidic upwelled waters. So I show you this just to really point out that we are the canary in the coal mine right now for this process of ocean acidification, and we're already starting to see the effects. Now, to try to put this into context of like what's going to happen in a kelp forest ecosystem, I'm going to talk about the chemistry. You don't have to memorize this chemistry, and if you're like, oh my gosh, I haven't seen a chemical reaction in 30 years, that's okay. I'm going to tell you what's important here. Okay, so the carbon dioxide reacts with water, that forms carbonic acid, and it dissociates into these two molecules that I have on the right. That hydrogen ion with a positive plus sign, that is what makes the water more acidic. It's what gives ocean acidification its name. Now, the problem is it's not the only reaction that happens with acidification. And that positive charge on the hydrogen ion attracts other molecules that have negative charge in the ocean. And in particular, it attracts these carbonate ions. So that's the CO32 minus on the bottom of the screen. So it attracts those carbonate ions, which react together to form more bicarbonate. Okay, again, details are not important. What I want you to see here is that we're seeing threefold changes in the chemistry of the ocean. And each of these changes has really different biological implications. So for example, organisms that build their shells or their skeletons out of calcium carbonate, carbonate is the key thing there, need that carbonate molecule. So as it gets eaten up by reacting with the hydrogen ion, we actually see lower calcification rates in things like abalone, things like mussels or sea urchins, coral reefs. And so a really wide range of species that build shells of skeletons out of calcium carbonate. What's interesting though, is that not everything suffers from acidification. So that increase in the bicarbonate ion concentration, that's this one here, HCO3 minus, actually benefits seaweeds and phytoplankton, or at least some species, who use bicarbonate as the starting substrate for photosynthesis. So we actually expect some of these seaweeds to actually benefit because they like carbon, right? That's what they need to photosynthesize. However, changes in that hydrogen ion concentration can cause really a wide range of effects. So changes in the acid-base balance or the intercellular chemistry of different species, as well as behavioral effects. And I don't have time to go into all of those. I show you this because it kind of points out the wicked problem of ocean acidification is that we're dealing with a really wide range of effects. And it is challenging to put these back into these really complex functioning ecosystems that are made up of seaweeds and fishes and invertebrates and whatnot. Now, not that we don't have some understanding of what these emergent effects are likely to be at the ecosystem level. So most of our understanding of this comes from these natural volcanic carbon dioxide vent systems. So this is a system that I've worked in for about 12 years now. These are shallow water, cold carbon dioxide vents that release primarily carbon dioxide. These are found in the Bay of Naples and Mediterranean. And rather than show you a whole bunch of figures, I'm just going to show you a couple of photos side by side of what the rocky reef looks like in ambient conditions near the vents versus the areas that are impacted by the vents. So this is primarily being caused by the carbon dioxide and gives us this natural laboratory to try to understand what global acidification might do to the reefs. And you can see, we see really clear changes in the diversity, reductions in the abundance of those species that build those calcium carbonate shells, and this dominance by fleshy seaweeds that can use that carbon dioxide in the system. Now, what's interesting here is that we actually think part of this shift is being driven by interactions between the species. So it's not as simple as some species can survive and other species can't survive. It's more about some species grow a little bit more slowly, some species feed a little bit more, and the changes in those interactions in this complex ecosystem can drive the shifts. So the example that I'm showing here is also at those carbon dioxide vents that I've worked at, where what we think is happening is that the sea urchins are not grazing down the system in the same way. So on the left, in ambient conditions, what the sea urchins do is they actually graze down the fleshy seaweeds and they create these bare spaces for more rare or competitively inferior species to colonize and maintain the diversity in the ecosystem. In the low pH conditions, they're not doing that in the same way, even though they're in the system. And instead, those places that they used to clear are now being dominated by fleshy seaweeds. So my lab, part of what we've been doing most recently is really focusing on these interactions between those grazers and the fleshy seaweeds, in part because we know they're really important for kelp forests. So Raphe just showed some interesting results showing that sea urchins in particular play a really important role in determining whether we have the kelp or whether they eat it all up, as well as the importance in these natural carbon dioxide vent systems and seeing those grazers. Okay, so that's the background. What I hope you get out of that is just that ocean acidification is this really global problem. It's happening everywhere, but we're really localized seeing the impacts of it already. And that we think that grazers might play some really important role in driving these ecosystem shifts that we think may be coming. Okay, so now I'm just going to tell you a couple of little stories of some research we have going on. This is about this very culturally important species of Pinto Abalone. And the story takes place in Southeast Alaska. So we started working in Southeast Alaska in about 2015. And we started working there because it's the very farthest northern reach of the kelp forest, or giant kelp, which is the species that we have off our coastline here in Monterey Bay. And we know that these colder conditions has more carbon dioxide in it. So we just wanted to see what was actually happening in these very northern kelp forests. And here's kind of the first snippet of data that we actually collected in the year 2015 and 16. I'm showing you the temperature in red and the pH in blue. And so you can see there's this very strong seasonal cycle. So the peaks in red are summer and the lows are the winter conditions. But what captured my imagination were these pH conditions that we were seeing in winter. So we were seeing conditions down as low as 7.8. So this is comparable to what we expect, again, at the end of the century in many other areas of the world for a long period of time, 3 or 4 months at a time. And we were really interested in what are organisms doing in these very low pH conditions? Can it give us some insight into how species might cope or their vulnerability to future changes? So again, I mentioned this story is about the Pinto Abalone. It is very culturally important. It's also the last recreational fishery for abalone on the US West Coast right now. Everything else is closed down because they have either been overfished or heavily impacted by these big marine heat waves. And what we think is that these lower pH conditions, or these more acidic conditions, are likely to increase their energetic demands, what they actually need to survive or to build that shell in these conditions. Importantly, this research was actually, the experiment I'm going to talk about was led by an undergraduate student in my lab, Cassie Powell. I'm showing there with a big old blade of seaweed, as well as contributed to by Emily Donham, one of my graduate students. Now, the other really cool thing about Southeast Alaska is we see these really striking differences in what's happening in the kelp forest in winter and summer. So this is a photo of me actually on the same transect in winter and summer. And you can see just by the color of the water, how much more photosynthesis is happening during the summer versus the winter. We think this is primarily due to light. But the thing that it brought up for me was that there's a lot less seaweed in the system at this window when we think, or during this window, that we actually think the abalone are going to need more energy to deal with these more acidic conditions. So we ran a quick experiment where we exposed Pinto abalone to current summer and future winter pH conditions. We then gave them a diet that would that they would be able to access during the summer months versus the seaweed that's actually available to them during the winter, which is the dominant Macrocystis chlorifera, or the giant kelp species. I'm going to jump right into the results. So I have growth on the y-axis, and then those current summer, winter, and future winter pH conditions on the x-axis. What we found is that acidification does indeed impede their growth, so there is an energetic cost associated with being in these low pH conditions, even in the current winter conditions. But what was really interesting is when we actually limited their diet to what they had available to them during the winter. So this is the ecologically realistic experiment, they're actually losing mass in those future winter conditions, suggesting that their populations are actually quite vulnerable to what might be happening in the future. So the thing I hope you take home from this is that we actually think that in these more northern or southern polar climates, that winter may be a really important window of vulnerability for herbivores because of the lack of the seaweeds in the system during the time when they need more energy. Okay, now briefly, I'm going to talk about one more example of some research. Again, this is being led by my graduate student, Emily Donham, about the Red Sea Urchin. And this research actually builds off of a collaboration with Yui Takashita in Jan Frewald, where we have been trying to actually get pH sensors and oxygen sensors into our marine protected areas across the state of California. And I'm showing 6 of those sites here. And then a little video of Emily Donham and Kate Violet actually deploying one of our sensors in a kelp forest. And what this research has shown us is many things I don't have time to go into, but I'll just talk about one example, is that in California kelp forests, low pH exposure is going to be associated with upwelling. So this is what I told you a little while ago, and I spent a fair amount of time talking about this. So I've got 3 figures here. I've got the pH, the temperature, and the dissolved oxygen concentrations over the same time. And you can see that those 3 graphs look exactly like each other. What that means is that when it's low pH, due to upwelling, you're also going to see lower temperatures and lower dissolved oxygen concentrations. So Emily ran an experiment that's really quite interesting, focused on red sea urchins. Now these are commercially harvested species. They play a very important role in kelp forests, as well as our local economies. And she wanted to know, how do they actually respond to low pH, low dissolved oxygen, and low temperatures? Because that is how acidification is going to show up in the California current. We think that it is still going to increase their energetic demands. It's just going to make it harder for them to survive. But what we want to know is like, well, do they actually then increase their feeding rate? Because they're in these conditions pretty regularly. So maybe they have some capacity to cope. So she designed an experiment where she actually mimicked upwelling. And I don't have time to go into it, but that's this little figure down on the right. So she created low pH, low dissolved oxygen, low temperature conditions. And here are her results. So what she found is that when you increase the upwelling, so that's what I have on the x-axis, as you move to the right, we're moving into more acidic and deoxygenated waters. And I have growth on the y-axis. There's a very clear relationship that the growth goes down. So this tells us, yes, there is an energetic demand associated with upwelling and more acidic and low dissolved oxygen conditions that impacts their growth. When we look at their feeding rates, which is what I'm showing you now, it's the same thing. Upwelling is on the x-axis, but their actual feeding rates are on the y-axis. It looks very similar. So they decrease their feeding rates, and this is based on whether they're in those conditions 1 month, 2 months, or 3 months. Now, upwelling happens quite, as you saw from that other figure, it's really quite dynamic through time. So the other thing we wanted to know is like, well, what if they just get exposed to it for a few hours or a few days? Then how do they respond? And the results look very similar. Their feeding rate also declines whether they've just been exposed to it or they've been in it for 3 hours or they've been in it for 3 days. So what this means is there's very little signs of like behavioral compensation. They're not dealing, because they have increased energetic demands, they're not actually increasing their feeding rates, even though there might be kelp in the system. So again, what does this mean? I think what it likely means is that we could see population declines associated with the red sea urchin, but also that upwelling in and of itself, while it does benefit marine organisms and it does drive the productivity in the kelp forest, may also be a window of vulnerability for those species that are more vulnerable to acidification. Okay, so coming back to these 3 themes, what I hope you get again is that ocean acidification is this global problem, but that has a very local impact here in California, and we're really at the front lines of these changes. That these seasonal dynamics or these variable systems where winter and upwelling may actually create these windows of vulnerability for marine species in kelp forests. And thus far, we don't have a lot of evidence that species are actually responding behaviorally to actually cope with increased energetic demands. So our next steps in the lab are really trying to look for the areas of adaptive capacity. So are species from areas that have more upwelling better adapted to deal with the changes in the future? Or are there other mechanisms of resilience that we can look to actually protect our kelp forests from these future changes? So I have lots of people to thank that contribute to our work. Our work is really challenging, diving in the winter in Alaska and along the California current. So a lot of people contributed. And then I also wanted to call out Michael Langens and Kate Violet, who contributed a lot of the beautiful photos in this, who were both undergrads at UCSC and were trained through our scientific diving program and graduated from the Ecology and Evolutionary Biology department. And with that, I will turn it over to Mark Carr, and then hopefully we have some time to answer questions.
Great, thanks, Kristy, and thanks, Raphe, as well. So we've received so far a handful of questions. So if you do have questions, feel free to continue to add them to the question and answer frames. But I'll go ahead and start. And the way I want to do this, I think, is to consider the questions chronologically, starting with Raphe and the issue of harmful algal blooms. The first question is actually rather technical, Raphe, and it was, you were showing these graphs of the probability of the particulate domoic acid exceeding 500 as a threshold. What is the basis of that? Is it based on any clinical outcome like toxicity to humans or other organisms?
Yeah, that's a great question. At 500, that's when we start to see it accumulate in shellfish and other things that are going to transfer that toxin into the rest of the food web. And so that's sort of our bottom number, where at 500 and above, there's the potential for consequences to organisms. And one of the things you always have to be careful about is we can measure much lower than 500, but it's probably not very relevant to a problem. And so we set that threshold to be conservative and say, when it's 500 or above, you're going to start having problems.
Right. And what I'm going to do is go down through the questions that we've received, and then, of course, I have many questions that we can return to at some point, perhaps. There was an interesting question about the satellite technology, Raphe, and this one, interestingly, asked whether there's any indications that the actual cumulative or repetitive scanning of the Earth by these satellites and other electromagnetic fields might contribute to ocean warming.
That's an interesting question. The satellites that I was referring to specifically are what we call passive sensors. And so it's like a camera where it's just simply measuring the radiation coming back from the Earth. There are active sensors that actually use microwaves. And so it makes sense that, we're blasting the Earth with microwaves from the satellite. Isn't that doing something? But they're so little energy compared to something like solar flare or other natural variability that you'd have to have the satellite like in your lab blasting a glass of water to actually see it heat something up. And so it's not having any significant impact on the oceans.
Okay. And now transitioning to your description of the loss of the bull kelp forest up on the north coast of California. So one of the overarching questions, of course, is there any indication or likelihood that kelp will recover to those pre-marine heat wave levels of the past?
That is a controversial question. There is still kelp there, and so it is possible for it to recover as long as there's, kelp to spawn and to survive. They could come back. In some parts of the world, once you get into an urchin barren, it seems to be pretty stable and it's really hard to get it to go back. But there are examples of it reversing. And so, particularly if the temperature keeps going up, urchins generally don't do well above about 24 degrees Celsius. And so you could imagine, for example, you warm enough to kill all the urchins and then it cools back off and allows the kelp to come back in. But that's really, as I said, previous El Niños, we still have kelp. And so they've been able to weather these big warm events without going to urchins. And in other places, the urchins have gone away. But it is concerning that there are many places around the world where the urchins manage to hang on for a very long time once they've established themselves.
Yeah, and in fact, there's a couple of questions that were related directly to potential mechanisms of recovery. One that you just touched on, which was whether these urchin populations would be expected to decline eventually because of the lack of kelp that they've created.
Yeah, you would think so, but they're hardy little buggers, and so they can go a long time and not die from starvation, which is surprising unless you work on them, and I'm sure you think it's perfectly normal. And so they've got mechanisms to hang out for quite some time. The other obvious one is, well, what eats urchins? And so sea stars, but they also declined because of the wasting disease. And then historically, we had otters going all the way up the West Coast. And right now, the current range stops in Monterey Bay. And so there's not another obvious predator. There's some really nice work that came out of connection to UCSC. Brent Hughes has a paper showing that if we recolonize San Francisco Bay with otters, we could actually triple the existing population. And so, if you wanted to do something like that, where you step in and allow the otters to expand back into Northern California, that would be a potential way to bring the urchin population back down. But right now, they're not there.
Right. Yep. And so you just answered actually a couple of additional questions, which were how the otters might be affected by the loss of kelp, how they might contribute to bringing down urchins. But the fact is that on the North Coast, they're just not there, huh?
Exactly. Yep.
Yep. Another question is more related, I think, to Southern California kelp forests, and whether yourself or Kristy want to respond to this. There was a question about sargassum invasions into the kelp forests in Southern California and reducing kelp. What's the geographic extent of that, and sort of the role of sargassum in this?
I can take a quick stab at that. So we have a lot of sargassum down in Southern California, especially in the kind of Santa Barbara area of the coastline. And it does actually, or can, outcompete the dominant kelp, the caniform kelp, and then create these very different ecosystems. We have had reports of it along the coast, the Big Sur coastline, from divers, as well as some reports in Monterey Bay, but it has not taken hold or really invaded the ecosystem here yet. But there's a lot of concern about what it might do to our ecosystems as well, and whether it might actually benefit from warming and acidification as well. So that's a really open question that people are quite interested in. And Raphe, I don't know if you have more you want to add.
Yeah, no, I think in terms of California, that's a great summary. I'll just comment that sargassum is becoming a problem globally. And so we're seeing sargassum showing up in a lot of places that we didn't see it before. And it's getting a lot of attention. And it's another one of those where, it's pretty clearly linked to changes in the ocean. And so it's another one of the winners, unfortunately, that are disruptive to other ecosystems.
Yeah, I would even add that at the carbon dioxide vents that I worked at, there are very few species that could live in the very acidic conditions that aren't even that reasonable to think of as future ocean. But sargassum, not the species that we have here, but related species of sargassum, was one of the few that can actually thrive in those really challenging conditions. So they're pretty weedy.
So before we shift to ocean acidification, a question for the two of you again, which is, what are the larger implications of the loss of these kelp forests?
I think Kristy could probably tell you lots of them, but some obvious ones are the loss of biodiversity. We love our kelp forests, or I love our kelp forests. It's a habitat for many, many, many types of organisms. A lot of juveniles use the kelp forest before they get large enough to leave. And there's something called blue carbon, this idea that naturally occurring marine organisms can help to mitigate climate change by buffering the ocean, raising the pH, and drawing down carbon dioxide. And kelp is certainly one of those. And so there's, if we didn't have kelp forests, it would fundamentally change these coastal ecosystems.
Yeah, and I would just add, one of the things that we've already seen on the North Coast due to the losses of these kelp forests is the closing down of the recreational abalone fishery. So it used to be one of the few places where you could still catch abalone in Northern California. This is the red abalone that we're talking about here. And that's just super culturally important for indigenous tribes, for people, locals on the coastline. It was really a big part of that community. And those populations have just been decimated because they rely on the actual kelp as food. And so that fishery has been closed. We don't think that fishery is going to open again. I have heard estimates like 2030 to 2050, way out there. We do not think those populations will recover very quickly. So that's just one example that has really profound implications for the economy also on the coastline.
Okie doke. Now, pivoting more to ocean acidification, one of the questions, Kristy, is whether you, and Raphe as well, whether you expect the upwelling patterns latitudinally from north to south along the coast to change as the diffuse ocean temperatures increase?
The model predictions that I have seen is that, so upwelling is a little bit more challenging to predict. It's a more local process that's, we don't have as great a predictive capacity for that. It's a little bit more like the weather, like we're good at predicting it at shorter timescales and it's harder to predict at longer timescales. But the models that I've seen generally predict that we're going to see an increase in the frequency of upwelling with global climate change. And I think that that's generally the consensus at this point. So that is actually maybe a good thing when we think about warmer temperatures, that these species, those upwelled waters are colder. They're bringing cold water onto the shelf. They will warm with warming as well, but it also brings that the acidification and then the deoxygenation up into that coastal zone. So how it plays out is not totally clear. Raphe, did you want to add?
Yeah, no, that's exactly right. A subtlety there is we have what we consider, our normal coastal upwelling, and then there's this upwelling caused by gradients in the wind, which is more related to offshore upwelling. And one of the things that we expect is there's going to be more offshore upwelling. And the reason that's interesting is the offshore upwelling doesn't bring up the same nutrient concentrations. And so you get a lot of mixing, but in particular, it's not bringing up as much iron as we have in the coastal ocean. And so as that upwelling changes, there may be these sort of follow-on effects where if you don't have enough iron, you select for different phytoplankton, and that means you select for different grazers. And so again, it's one of those things where a seemingly simple change can have these unusual feedbacks that end up changing the way the biology works.
So here's an interesting one. Starting with ocean acidification, but then also, Raphe, with respect to phytoplankton productivity, perhaps, all of these fires that we're having along the coast of California, the ash that's being transported offshore and deposited in surface waters, is there any chance that that's influencing acidification or nutrient supplement to plankton populations?
I'll let Kristy tell you about acidification. In terms of nutrients and the phytoplankton, we actually had this sort of coincidental opportunity to look at that during the Thomas Fire down in Santa Barbara. And we had a boat out there that was run by graduate students actually from UC Santa Barbara. And they had an instrument of ours that's basically an underwater microscope that takes pictures of all the phytoplankton. And they were able to go in and out of the ash plumes that were falling into the Santa Barbara Channel. And what they saw is where the ash was being deposited into the surface ocean, we had anywhere between between 10 times and 100 times more harmful algae showing up. And they did some really nice work showing that that ash was actually loaded with nutrients. And so you were actually selectively stimulating these algae that were there anyway, but they got essentially a boost of nutrients and took off because of that ash. There's a lot of other work going on right now because of our fires. And so I know there's many groups looking at that, and I'm sure Kristy can comment about what that does to acidification.
Yeah, actually, I haven't thought about it very deeply. So it's an interesting question from the folks who are watching. So I would say also that it probably more as a nutrient pulse to the system. What would actually buffer acidification would be what we call alkalinity, and that's more coming from like calcium carbonate, so dried shells or baking soda type thing. So the ash itself, I would think, would bring nutrients. The way that that could affect acidification, again, is by stimulating the phytoplankton, which use the carbon dioxide. So you could very locally kind of reduce the acidification signal, but that would be a pretty local impact associated with very short term.
So here's a question that you sort of alluded to, Raphe, but Kristy, I know you think about this quite a bit as well, which is with these species range shifts to the north, right? Because of these warming waters. What do you think the near shore ecosystems are going to look like here in 100 years? And good luck with that.
Well, if you're around in 100 years, you can tell us whether we're right or wrong. That's a good question. I mean, there's more going on than just climate change. And so a lot of it is human use of the coastal ocean. And so one of the big drivers, completely separate from anything going on with climate, is how much nutrient pollution we put into the coastal ocean. And California has been really good and proactive about minimizing that. And so, you know, we may have an ocean that looks pretty similar to today, other than some species shifts, if we're not careful about the combined effects of temperature and acidification and nutrient pollution. You know, we may look more like Florida, where they've got massive green mats of stuff floating around because it's just simply, it's warm and there's lots of nutrients and you selected for organisms that like low pH. And so hopefully we don't see that. You know, one of the, you know, sort of a positive thing is that California is an upwelling system. We naturally see these changes. We naturally see El Niños. And so there is a lot of resiliency in the organisms that live here. And in the next 100 years, you know, some of them are probably not going to make it, but most of them, I would expect, we're going to find that they're much more resilient than we might have thought.
I would just add one piece to that, which is this idea that a lot of times when I talk about the California current to people, I say it's not a gradient. I want you to think of it more like a patchwork quilt, like it's a mosaic. And so you can have areas of very low pH upwelling in Southern California and warm areas of no upwelling in Northern California. There is a general latitudinal trend, but within that trend, it's really patchy. And so it's, I don't think we're going to see, you know, like the whole thing just shift northward because of that patchiness. Now, I'm just totally, you know, guessing here, but what I think is that we're likely to see some shifts in the community, in that we will see some more Southern California species, but it's not going to be an entire shift. We'll have kind of patchiness in the ecosystem still.
Mm-hmm. There was also a question with respect to the depth range of these effects. So with changes in upwelling and acidification, what about deeper benthic ecosystems and seamounts offshore? Is anybody looking at that and the potential effects?
Yeah, absolutely. People are quite concerned about deep sea organisms. And in fact, that was a lot of the early research was on how these deep sea organisms might be impacted by acidification because all that carbon is kind of congregating in the depths of the ocean. Yeah, so those deep sea corals people are quite concerned about, and there's been research showing declines in their calcification rates as well. And yeah, in seamounts as well, people are quite concerned because essentially we have this depth that we call the saturation horizon. So kind of below that depth, it's hard for things to calcify, and above that, it's things calcify pretty easily. And what's happening is that's getting shallower and shallower, and so it's moving up these seamounts and things, and so it's just making it more acidic and challenging for organisms to calcify and grow in those conditions.
Okay. Moving more into the policy realm, Kristy, this should be deja vu. Here's a question saying, I'm wondering how you would recommend that MPA-related research, marine protected area related research, can advance appreciation for that intersection of the three areas that you laid out in the beginning of your talk. And what other key questions related to resilience should we be pursuing? And are MPAs enough, or you combine them with other management approaches to ameliorate or mitigate these impacts?
I think you have to think about other management approaches, but I think MPAs play a really important role, and they're one of our major management tools in the coastal ocean. So one of the things that we're working on right now is just trying to bring this understanding of environmental change to our marine protected areas. So as I mentioned, we have one of the best ecological monitoring networks of our MPAs of anywhere in the world. But we have very few sensors out there. We don't actually know what the chemistry is or the temperature is in our marine protected areas. So at a very base level, one of the things that we're trying to push for is just like, let's get some monitoring in these marine protected areas so that of the environmental conditions, so that we can better understand our analyses of like what the marine protected areas are doing. So for example, say you've got a marine protected area and you've stopped fishing in an area, but populations are still declining. Without actually monitoring the temperature or the environmental chemistry or the carbonate chemistry, you don't know, is that due to the fishing or to the stop of fishing, or is that actually a totally different thing? Is that global climate change? The other thing that marine protected areas can do for us is they can give us this, this is what a community or ecosystem, how it might respond when you don't have these other human pressures in the ecosystem. So I think that they can provide what we were calling these sentinel sites for understanding the effects of global change and how actually managing human activities may improve species responses to it. But I do think you have to take a variety of approaches to thinking about responding to global climate change in the coastal ocean beyond marine protected areas. Yeah, I could go on there, but I feel like I've been talking for a little bit already, but yeah, I think you have to think about it pretty broadly.
You know, here's a quick one, hopefully, and then I want to close with a couple of my own questions for you. This one is the devil's advocate, which is, hey, things change, get over it, right? The world's been changing forever. Species evolve, they adapt. So the alternative is to kind of not try to intervene with these efforts. And so what are the costs of that? What's the balance of not intervening?
I think there's a couple of things that I would say to that is that, yeah, I think you're right. In the really big global picture, things have been changing for a long time, but never this fast, and never when there were humans around that were going to be so heavily impacted by the changes. So I think our ecosystems are going to change and adapt and survive, but the impacts of these changes on human communities could be quite profound. And that's part of, you know, really our focus on it is not just for kelp forests, but it's for the human communities that are reliant on kelp forests for fisheries and tourism and all of those different kinds of things. With that being said, even if we stopped tomorrow, sorry. What I was just gonna say, with that being said, environmental change is coming. Whether we stopped emitting carbon dioxide tomorrow, we're still going to see changes. So we can't just think about mitigating. We also have to think about adapting. How are we going to actually roll with the punches somewhat and prepare ourselves so that we can work with the oceanographic changes.
Okay, so I'm going to close with two questions, one for each of you. And you only have 2 minutes each to tell your life story. How, because, you know, there's a lot of students in the audience. And so I'd like, if you can, to tell them how, Raphe, you became a plankton nut and so fascinated with harmful algal blooms. And you, Kristy, with kelp forests and ocean acidification and climate change.
All right, so the less than 2 minute version, I actually started as a pre-med student, and we got to the point where you had to do comparative physiology, and I wasn't into, you know, looking at dogs and thought that was not very nice. And so I ended up doing a summer REU program and went to coastal Georgia and looked at production of sugars in salt marshes and discovered I loved the ocean. And from there, you know, it's kind of the harmful algal blooms and phytoplankton was, you know, it's a super interesting question, and it's something that, you know, it's everywhere. It has direct impacts to humans, and what I like to tell students is, you know, what I love about my job is I can work on anything, anywhere, as long as I can find somebody to pay for it. And so, you know, it's fun. You know, it's great to actually address science questions that have a real impact and that you can make a difference.
Awesome. I was not a pre-med student. I was one of those people that at 14 decided I was going to be a marine biologist, and believe it or not, here I am. And I was actually a UCSC undergrad, and that's what brought me to the kelp forests. So I came to the University of California Santa Cruz thinking I wanted to be a marine biologist. I got introduced to kelp forest ecology and just loved it. I loved being underwater and actually getting to experience these ecosystems. I took a little bit more circuitous path to where I am now. And I would say what really captured my imagination was being able to work on questions that I thought were really important for human society. So when I had the opportunity to do research on climate change and work in policy and management and try to contribute to solutions, it really caught on for me. As well as being able to dive underwater for my job. Is a pretty cool thing. And then getting to work with students is about the most fun thing in the world because they bring all of this energy and enthusiasm and passion and new ideas. So.
Absolutely.
Right. Hey, you too. Thanks so much for sharing, for the presentations, for sharing your thoughts about all of this. And George, I think I'll turn it back over to you now.
Thank you, Mark, Raphael, and Kristy, and thank you all for joining us tonight. And please be with us for our next crawl lecture, Cultivating Leaders and Solutions for Coastal Sustainability with Professor Ann Kapuscinski on March 18th, 2021 at 5:30 p.m. That's 5:30 p.m. California time. See you later. Take care, everyone.